so the meninges are the protective coverings. So the most superficial one, which would be directly adhered, if we're talking about in the brain, directly adhered to the underside of the cranial bone, right? So this would be where the scalp would be, okay? And then you have our compact bone, spongy bone, compact bone. These would be flat bones of the cranium. On the underside, this fibrous connective tissue, this is called the dura mater. So the dura, think dura, durable, mater, it's like mother, protective coverings, mother. And so this dura mater is the densest, it's dense fibrous connective tissue, and it just adheres to the periosteum on the inside surface of the cranial bones. And it's really just a thick protective covering, right? So this is the dura mater. Now, there are areas of the dura mater that can actually split into two layers and form these structures called the dural sinuses, and I'll talk about what that is in just a second, but it's a venous, it's a part of the venous blood flow system, venous blood. Now, directly adhered under the dura mater, this white layer would be, this is the arachnoid mater. An arachnoid, because it has a spider web-like appearance, these fibers come down from the arachnoid layer and connect to the pia mater, which is the innermost meninge. Now, the arachnoid mater is not vascularized. The dura mater is. It contains blood vessels. The pia mater is vascularized. The arachnoid mater is not. Now, these structures that are projecting into the dural sinuses, these are called arachnoid villi. Okay, arachnoid villi that protrude. They bulge into the dural sinuses. Now, there is a space. Here's the arachnoid mater, this white line. Then there's a space called the subarachnoid space. This is where the CSF flows, the cerebral spinal fluid. Now, this pink line or red line that is directly adhered to the brain itself, this is the pia mater. And the pia mater is so directly adhered to the brain that it's almost impossible to tease apart. So when you touch the brains in lab, what you're touching is actually pia mater. Very highly vascularized. It contains all the capillary beds that will nourish the cells of the brain. And so we have dura mater, then we have arachnoid mater, then the subarachnoid space where CSF flows, and then the pia mater, which is a very delicate connective tissue directly adhered to the brain. It follows all of the contours of the brain, and it's highly vascularized, contains all the capillaries. So in the flow of CSF, when you watch the video of me going through the brain, the function of the CSF is so the fact that there's a subarachnoid space and it surrounds the entire brain and spinal cord, it essentially allows the brain and spinal cord to somewhat float. It's floating in the CSF, so it gives it a buoyancy, especially with the brain, so that it can still be very dense but not weigh down. So it creates buoyancy. It also acts as a shock absorber to any blows that you might get to the cranium because there's this fluid here. It absorbs shock. It also plays a nutritive function because it's the capillaries in the ventricles that push out or produce CSF. They will contain certain ions and certain chemicals, certain things that can help with nourishment. It also acts to remove waste. So as the cells of the central nervous system produce metabolic waste, they get dumped into the CSF, and CSF is constantly being recycled. We make more every day. Throughout the entire day, we're making new CSF, and old CSF that has a bunch of waste products will actually get reabsorbed through these arachnoid villi, get dumped into these dural sinuses, and then it will return to venous blood flow to be recycled. And then the ventricles in the brain will produce more, will flow through the subarachnoid space, and it will fill with waste, get reabsorbed by these arachnoid villi or arachnoid granulations, go back into venous blood flow through these dural sinuses, and be recycled. Okay?